Thank you. First of all, I would like to thank Jyoti for providing this excellent learning platform for all of us. So the abstract I'll be presenting is by Nabil Eshrafil et al., who studied radiomics model based on magnetic resonance imaging compilation, or the MAGIC sequence, as early predictor of pathological complete response to neoadjuvant systemic therapy in triple negative breast cancer.
Can you please make it full screen?
I'll just do it. Wait a second. Is it visible now? Full screen?
Not.
I'm full screen. I've put it on full screen only.
You have to go into the presentation mode first.
Okay, just a while.
Otherwise, it's okay. Your slides are well visible, so we can go ahead.
Okay.
Yeah, perfect.
So the background of the study, triple negative breast cancer is comprised of biologically aggressive tumors with diverse clinical behavior and response to chemotherapy. Prediction of disease response to neoadjuvant systemic therapy is critical to development of personalized medicine in TNBC. So in this study, they basically studied a novel MRI sequence, which is a magnetic resonance imaging compilation, of the MAGIC sequence, which is capable of simultaneous quantitation of several tissue properties, including T1, T2 relaxation times, and proton density, which may be reflective of tumor heterogeneity. And radiomic signatures from T1, T2, and PD quantitative maps may play a useful role for treatment response evaluation in TNBC patients. So objective of this study was to evaluate the ability of a radiomic model extracted from a novel MAGIC MRI sequence acquired during NAST to predict pathological complete response in TNBC patients. So the patient selection included 184 women, 122 in training dataset and 62 in testing dataset, who were diagnosed with stage 1 to 3 TNBC and enrolled in the IRB approved prospective clinical trial. All patients received 4 cycles of anthracycline-based chemotherapy, followed by 12 weeks of taxane-based therapy, followed by surgery. PCR was defined as absence of invasive carcinoma in the breast and nodes at surgical pathology. So they studied several features. The imaging features were studied. All patients were scanned with MAGIC sequence on a 3T MRI scanner at first of all at baseline, and then after 4 cycles of chemotherapy. T1, T2, and PD maps were generated from the source images using a software, which is a synthetic MRI. Textual analysis was done later on. Tumor regions of interest were segmented by a breast radiologist at T1, T2, and PD maps using in-house developed software. And 310 textural features were extracted from each map. So a total of 930 features were studied for each patient. So this is the image showing the T1, T2, and PD quantitative maps which were generated by the MAGIC MRI sequence. So once the source images were obtained, this was followed by extraction of T1, T2, and PD maps from the MAGIC sequence, and segmentation of ROI was done on each map. This was followed by selection of the volume of interest from each map and extraction of radiomic features. And then feature selection analysis was done to build the best model for PCR versus non-PCR prediction. Radiomic features were compared between PCR and non-PCR using Wilcoxon rank sum test. The area under the receiver operating characteristic curve was used for univariate analysis to determine textural features predictive of PCR. Logistic regression was used for multivariate analysis for feature selection and building the radiomics predictive model. And the model performance was assessed by leave one out cross-validation method. So in the results, they found that on univariate analysis, 35 PD, 42 T1, and 98 T2 radiomic features at C4 time point were able to predict PCR status with area under the curve more than 70% in testing and training cohorts. The top performing radiomic features were entropy, variance, homogeneity, and energy. On multivariate analysis, they found that multivariate radiomic models from C4 PD and C4 T1 maps showed best performance during both cross-validation and independent testing. The radiomic signature of C4 T1 map that included 27 features had best performance with area under the curve of 0.77 and 0.70 in training and testing cohorts respectively. While on C4 PD maps, there were 6 features were able to predict the status with area under the curve 0.73 and 0.72 in training and testing cohorts. So this was a multivariate radiomic model which was created for C4 T1 and C4 PD maps. So basically they concluded that this sequence or the magic-based radiomic signatures could potentially predict pathological complete response in TNBC during neoadjuvant systemic therapy. This data also shows the potential application of magic radiomics model for improvement of response assessment in TNBC. This was an example. This was a patient who was included in the trial. So these are the images. These are the initial images at baseline, followed by the images at C4 and at the after completion of 4 cycles, and followed by pre-surgical assessment by MRI. So the pre-surgical radiomic model based on C4 imaging predicted a non-PCR, and as we can see that the pre-surgical imaging showed residual disease, and this was confirmed by pathology at surgery. So this was quite an interesting sequence. So although we know that there are certain features like peritubular edema and central necrosis that are associated with worse recurrence-free survival, but these new MR sequence will definitely help in understanding the tumor heterogeneity in patients with TNBC and more targeted and personalized therapies. Thank you.